All right, so in the last section we were talking about the Mendelssohn-Stahl experiment, and it was determined, of course, that the copying mechanism followed the semi-conservative mechanism, and what you had is that complementary base pairing between the two strands that were containing the information to kind of synthesize the other one. So take a moment here and look at the two strands that we have in purple at the bottom, and go ahead and write the complementary sequence to that. Okay. So if you do that, what you'll see is you know that with 5' end is always going to line up with the 3' end. And then if you did this, you'd see TGA, GCC, ATT, and then you'd have 5'. And then right here you'd have 5', and then you'd have ACT, CGG, TAA, and then 3'. And if you look at those, they're exactly the same. So you can see what we did basically was just split those two purple strands apart from that original parental strand, and then we gave rise to these two new strands, which are exact copies of the other one. Okay, so once the mechanism for DNA replication was worked out, the details of how this actually worked became the topic of interest. Okay, and what happens is you have an origin of replication, which is where DNA replication actually begins. And in bacteria, you've got one of them, and there's hundreds of thousands of them in the eukaryotic chromosome. And that's because there's so much more DNA in a eukaryotic cell than there is compared to a bacterial cell. You'd never have enough time to get it all done if you only had one origin of replication on each chromosome. So there's multiple ones, okay? So if you look at this electron micrograph, you can see how this circular piece of DNA is kind of being replicated. So what you would have basically in one spot, okay, these were formally, say, joined together. That's where the origin of replication is working. You can see by these arrows here kind of working in the opposite direction, and eventually it gets to the end, and then you end up with two chromosomes. And if you recall something we talked about earlier in the year with John Cairns and the experiments he was doing with the radioactive tracers to basically see how DNA was replicated, if you look here, you can see here's kind of it's moving in this direction, so to speak. So it's basically peeling apart, much like you're seeing right here. And this is what's giving rise to two new chromosomes there. And again, this other one, this is just from an earlier slide there, just seeing that the DNA of a bacterial chromosome is actually circular. Okay. So with DNA replication, there's a complex system of enzymes that are involved, and we're going to go through each of these here in terms of what they do. So you have helicase, DNA polymerase, topoisomerase, which is also referred to sometimes as DNA gyrase, primase, DNA ligase, and then some single-strand binding proteins. So here's just a quick video that I want you guys to watch, and then we'll kind of go through it, and then we're going to come back and watch this again at the end, and I'll be more than happy to go over it with you guys in class if you have questions about it too. Okay?
DNA is replicated by continuous synthesis of a leading strand and discontinuous synthesis of a lagging strand. Coordination between leading and lagging strand synthesis is achieved by the dimerization of DNA polymerase molecules at the replication fork. The DNA polymerase dimer moves with the replication fork. The polymerase at the leading strand template remains attached to the DNA, continuously synthesizing the leading strand. The lagging strand polymerase initiates DNA synthesis at the fork from an RNA primer made by a primase complex. The polymerase elongates the lagging strand in a direction opposite the fork, but stays bound to the fork. As a result, the newly synthesized lagging strand fragment loops out between the polymerase and the fork. Once the polymerase completes an Okazaki fragment, it dissociates from the DNA template. A new primer is produced at the fork. The polymerase reassociates with the template at this position to continue synthesis of the lagging strand. By this mechanism, the two polymerases can add nucleotides to the growing strands at the same time, and at rates up to 1,000 base pairs per second.
All right, so if we look there, it was 1,000 base pairs per second. That's in a bacterial cell. And in a eukaryotic cell, it's about 50, I think, base pairs, about 50 base pairs per second. There's some details there that you don't need to worry about, but we're going to go through these. I just wanted you to kind of see it so you have an idea of what you're looking at, what we're talking about, and then, you know, we'll watch it again, of course. We'll watch it in class, and then I'll go back through it with you guys, okay? So we're going to kind of go through some of the details of this. So if we're looking at this here, hold on one second. All right. So now when we look at the helicase, what the helicase does is it basically pries apart the double strands. So as you have like 2 strands of DNA right here, and they're joined by these hydrogen bonds, of course, you have DNA helicase that's moving in this direction, kind of unzipping the DNA. So it's separating them and making them available so each one of the 2 strands can be copied. And then out ahead of helicase, you usually get kind of the over twisting of the DNA, and you wouldn't want to break the strands or anything like that. So there's this enzyme called DNA gyrase or topoisomerase that's kind of acting to stabilize the DNA to prevent breakage out ahead of the helicase and the replication fork, okay? And then once the DNA gets separated, now you've got these single strands, and these single strands have the ability, as you see down here, where they can kind of loop back on one another and hydrogen bond, and that would cause all sorts of different types of problems with this replication. It would prevent it essentially. And so as DNA polymerase is sliding along in this direction here, what it needs is this kind of free, unbound single strand that it can add the nucleotides to. So in order to prevent this, to keep these things from binding on one another, you have these single strand binding proteins. There's a whole series of them. And what they're doing is they basically just attach on to the single strand of the DNA. They prevent it from hydrogen bonding to itself, and they enable the DNA polymerase to kind of slide along and add the nucleotides to it, okay? All right, so now you have an enzyme here called primase, and what primase does is it basically primes the pump. What it'll add to the DNA are some RNA nucleotides, and it's important to keep in mind that I know we're making DNA, but we're going to add some RNA primers to this. And these things are complementary to the DNA strand, and this is basically where the whole process kind of initiates. This is where it gets started. And these primers are these short fragments of RNA. They have an available 3' end, and it's important for you to remember that they're added to the free 3' end of this molecule. I'm going to go over that with you multiple times, but just remember it's added to the free 3' end. It's going to be important for a couple of different reasons, which we'll talk about coming up. And then you have DNA polymerase 3, and this is going to add these nucleotides according to the base pairing rule. So when it encounters an A, it's going to add a T, C, it's going to encounter G, and so forth. And primase gets this entire thing started, and then as the process of replication continues, what you'll have is DNA polymerase 1 going to remove these RNA primers and replace them with actual DNA nucleotides. And then once that occurs, you're going to have a kind of a continuous DNA molecule, double-stranded, but you're going to have some breaks in there where the RNA primers were pulled out and the DNA was put in. And then what you have is this enzyme right here called DNA ligase, and what that's going to do is form the phosphodiester bonds. It's going to join the gaps in the fragment together so that you have a continuous strand of DNA, which is what we're making, okay? And what you want to do is you want to keep in mind that all of the molecules that we're talking about here to kind of, you know, I guess describe the process of DNA replication. You're trying to maximize the efficiency, and you're trying to do so accurately. And all of these things that we're talking about, all of those enzymes and such that we listed there at the very beginning here, you want to think of those as a DNA synthesis machine, because what they're doing is they're synthesizing DNA, and they're all working together, and they're all making up a part of this complex machine that can't work without any of the individual parts. Okay, so that's going to end part 2. I'll see you about part 3 here in just a few minutes.